I'm going to talk about the impact of Pfumvudza on crop yields, weed density, weed species diversity, and soil quality across agroecological regions of Zimbabwe. Right. Conservation agriculture, which is CA, is a climate-smart cropping system that ensures high yields in agricultural environments exposed to frequent droughts. CA has three pillars. Number one, minimum soil disturbance, which is planting in basins or simply reaping. There should be permanent soil cover. Number two, then there should be crop rotation. Farmers have often failed to implement all of these three pillars. There are constraints which are associated to CA, which are weeds and poor soil fertility. Pfumvudza is the local name for CA. It also has three components: minimum tillage, soil mulching, and legume-based rotations. Now with the Pfumvudza, the plot size has been reduced to 39 meters and 16 meters. Probably this can facilitate operations. The plant spacing is 75 centimeters and 60 centimeters in row. For sorghum, it's 75 centimeters times 30 centimeters. The inputs, we've got 5 kg seed, 12 kg lime, 50 kg compound D, and 50 kg ammonium nitrate. The advantage is the timely planting. This is a slide showing the Pfumvudza system. You are showing somebody top dressing. You can see the amount of mulch is not that much. Now, one of the problems with the CA or Pfumvudza is weed problems. Now, these systems, in most cases, allow weed seeding emergence to go ahead of crop emergence, thus giving weeds a competitive advantage. It was found that weed biomass in conventional tillage compared to basins or reaped plots was higher. It was the presence of mulch which was responsible for reducing weed biomass. In contrast, earlier studies had shown that higher weed biomass actually occurred in conventional agriculture, in conservation agriculture, compared to conventional tillage. There are also studies which showed that the weeding frequency was 2.7 times in conservation agriculture compared to 1.7 1.9 times in conventional tillage. The differences in results could be accounted for by the presence of mulch, because mulch appears to regulate weed seedling emergence. So different researchers used different mulch levels. One of the problem weeds is Striga asiatica, which is a parasitic weed which can parasitize cereals, notably maize and sorghum. The problem with this weed is that it can damage the crop before it emerges. Even if farmers want to weed this crop, they cannot see it. They can just see the crop, which is wilting. By the time this weed emerges, the damage has already been done. The weed is associated with low soil fertility. The current hybrids are susceptible to this parasite, mostly in natural region 3, 4, and 5. This then is the Striga asiatica. This is in Gokwe. Imagine you put in your Pfumvudza in such a Striga hotspot. This again is the weed on a heavy soil. This weed can grow across many soil types. Here I want to talk about the effects of mulch, because mulch is key to the success of Pfumvudza. Now, mulch, the absence of mulch, like what happens on a bare soil, small seeds of weeds can interact with the light. They have got a phytochrome, which senses light. So basically, when you have light, phytochrome red is converted to phytochrome far red, which can trigger germination, causes weed seedlings, and then you get your weeds infecting the sites. No wonder why systems of conservation agriculture with less mulches suffered a lot of weeds. On the other hand, if you have mulch can create darkness, and this darkness will allow the conversion of PFR to PR and drive the weed seeds into seed dormancy. Thereafter, there is no seed germination and there are no weeds. However, seed size is something to do with this phytochrome system. Big weed seeds do not need the phytochrome signal. They can germinate in darkness. Mulch causes the following effects. It can cause etiolation or abnormal growth. Weed seedling is chasing light most of the time. you can have chlorosis, a lack of chloroplasts. There's no photosynthesis, no carbohydrates. There's starvation and death on these seeds. Some weeds could obviously escape through the mulch, but mulch can be effective. Whether the weed has got big seeds or small seeds. Small seeds through the light system. Big weeds through etiolation and chlorosis. Mulch can also possess chemicals which can kill weed seedlings. For example, sorghum residues. Here I want to focus on the weed seed bank dynamics so that we can appreciate the role of mulch. Now, if weeds are allowed to flower and produce seed, like what happens most of the time, they will add seed into the soil via seed rain. That's how we describe the process. Like during this period, we are expecting seed rain. There could also be seed dispersal, which is caused by the actual rain, wind, animals, or machinery. The weed seeds can be brought into the fields. Now, this is an external input, which you may want to call the foreign direct investment from other areas, as opposed to seed rain, which is domestic investment. Now, within the seed bank, you have got seeds which are on the soil surface and those which are within the soil up to 30 centimeters. Now, what can happen is that if we have mulch, it can prevent seed rain because it stops seed germination. If we have foreign direct investment or seed dispersal, the seeds are trapped in the mulch. They have no contact with the soil, so they are not going to germinate. Those seeds which are already in the soil may be dormant. But should some seeds germinate, like big weed seeds, they can suffer etiolation, lack of chlorophyll, and eventually die under mulch. So most of the seeds now remain dormant. And there's a period when these weed seeds remain viable. Blackjack tsine can actually survive for up to 3 years. After 3 years, then it's gone in this seed bank. But others can go up to 12 years, like Racine indica. The striker seeds can go up to 20 years. So does the reduce the size of the CA, which is now 39 meters times 16, increase weed control efficiency? Soil fertility problems. CA can be challenged or this Pfumvudza is going to be challenged by poor soil fertility status, where we have low nitrogen, phosphorus, and soil organic carbon, low soil pH, which is around 4.3. There's also a lot of leaching. Even if you apply the fertilizer, they can be easily lost. There is also lack of response to fertilizer application. So is Pfumvudza going to increase crop yields in poor soil fertility environments? There are some reports which have confirmed that actually under CA there are improved crop yields. The aim of this project is to assess the impact of Pfumvudza program on crop yields, weed density, weed species diversity, and the soil quality parameters across agroecological regions of Zimbabwe. The objectives are to establish weed density and weed species diversity under Pfumvudza compared to conventional farming methods. The second objective is to determine maize, sorghum crop yields on the light, medium, and heavy textured soils in different agroecological regions. The third objective is to determine the effect of the Pfumvudza on selected soil quality parameters, for example, soil organic carbon and the pH, and the soil nutrient status. Now, how are we going to do it? We are going to conduct diagnostic surveys to gather some information. These diagnostic surveys will be done in natural region 1 up to 5. Probably at this stage, I want to highlight that Mazoe and Makonde are actually natural region 2. We need sites which reflect region 1. We are going to collect information on crop production practices for Pfuvudza plots and also plots under conventional farming methods across these 5 agroecological regions. Probably the target sample could be around 40 to 50 farmers on the light, medium, and heavy clay soils for each site. Weeds, crops, and soil sampling will be done in these 5 agroecological zones. There will be 2 sites which will be sampled per agroecological region. Now we'll have a questionnaire which will enable us to gather information from the Pfumvudza plot as well as the conventional tilled plot. Our questionnaire will target information on weed information. We'll ask farmers about their problem weeds, also sample. We need to know soil type, the previous crop, current crop, planting date, crop emergence problems, fertilizers, basal and top dressing, which were used, cattle manure, lime. Was there any soil analysis done? Then mulch type, weeding frequency, did they use some herbicides? Then on the studies themselves, we are going to do a weed emergence study. This study involves sampling weeds using 45 times 45 centimeter quadrats. We'll actually count the weed species and record them. And the following parameters will be estimated: weed density, weed species richness, weed diversity, weed evenness. So this is the field where Pfuvudza is done. This is how we are going to sample 5 positions using quadrats. We'll also do a weed seed bank study. This study will give us an estimate of the number of weeds or weed populations in Pfumvudza versus in conventionally tilled plots. What we missed in the first study, we can always get some information on the weed seed bank study. We'll sample soils 0 to 10, 10 to 20, and 20 to 30. Now, for each depth, we will have to composite the soils. These soils will be put in shallow trays to encourage germination and weed seedling emergence. Once every 2 weeks, the weed species are recorded, and this can run from August to October. This will give us estimates of weeds which were added this season into the soil seed bank. Probably we will have more seeds in the conventional tilled plots versus the Pfumvudza plot. Then on the soil quality parameters and crop yields, crop yields will be estimated in the Pfumvudza plots on light soils, medium and heavy textured soils. At the same time, soils will be sampled 0 up to 20 centimeters, and these soils will be subjected to nutrient analysis in the laboratories. We will focus on soil carbon, pH, exchangeable bases, cation exchange capacity, total and mineral nitrogen, and phosphorus. Now, on data analysis, the information on the weeds will basically compare Pfumvudza and the conventional tillage using a t-test. We'll also create some tables which show weed species distribution. We will be also interested to find out whether climate change has had an impact on weed species shift. What we're going to do, we will use the weed survey done by Chivinge. It was a national weed survey in 1980 as the baseline, and see what has now happened between 1988 and up to this last season. use or calculate percentage. We will have the number of weeds similar to the Chivinge 1988 National Survey divided by the total number of weeds of current weed survey, for each site times 100. Soil analysis quality parameters and crop yield. Basically, ANOVA will be used to compare within each agroecological region and between agroecological regions. And LSD will be used to facilitate comparisons. I should think that there will be a stage where we are going to see or examine associations between weeds and the soil fertility at some point. The expected outcomes. This approach may enable us to identify mulches which are allelopathic to weeds. This could address the problem weeds in Pfumvudza. If there are serious striker hotspots, there are opportunities to target maize hybrids, AX28 and AX31, which were bred specifically for striker resistance or tolerance. Here to use it. Probably this will give us an opportunity to further evaluate these genotypes. We could identify legumes which could be useful, which could be used to improve soil fertility. We're also thinking that some of these legumes could have allelopathic effects to weeds. Who knows, we may end up recommending what are known as organic herbicizers, materials which can kill weeds and also improve soil fertility. Now, in order to address the problems of soil pH and leaching, we may have to borrow from Professor Gwenzi, who used the biochar to regulate soil pH. Maybe we may end up putting biochar in these planting basins to stabilize the soil pH. This is quite an intensive work plan. We will start from June and ending up in November with the production of some thesis. We've got the Department of Soil Science and Environment who are participating in this project. We've got Professor Uta, who is actually leading the whole group. And we've got Dr. Nezomba, Dr. Jairinda, Mr. Nyamgafata, students. Then we've got plant production sciences and technologies, Dr. J.T. Rugare and myself also following with our students. We've got Madzimure Innocent, Kudanga Moses, Viola Manyota, Ian Kasoni, and Nyarai Betera. Thank you.